Hey everyone, welcome back to the channel. Today we are breaking down one of the absolute core concepts in pathology that you must master for your NEET MDS or BDS second year exams, and the topic is reversible cell injury. So I'm going to make it super simple, high yield and easy to remember. So make sure you grab your notes and let's jump right in. So look at our first page, that is reversible cell injury. So what is the core concept behind it? So think of reversible cell injury like a rubber band. You stretch it with stress and its shape changes na, but the moment you let go it snaps right back to the normal. So that is why its core concept is a state where cell undergoes functional changes and to a noxious stimuli in response to a noxious stimuli, but can return to normal if the stimulus is removed. Okay, and look at this green box, and the key rule is the cell is injured but not dead. The injury is sublethal, meaning the structural integrity of the cell is still maintained. Okay, so let's talk about the causes, matlab what hurts a cell. It's etiology. So etiology is first में hypoxia, ischemia most common cause है, and which is less O2 means less ATP production. एंड इसका एग्जांपल ले सकते हो कि इफ़ द पावर प्लांट जैसे गोज डाउन, या कोई भी सिटी में कोई पावर प्लांट है और वो बंद होने हो जाता है, तो सिटी भी वर्क करना बंद हो जाएगी, क्योंकि वो उसका मेन सोर्स ही सिटी का जितना भी है वो पावर प्लांट से है. तो अगर पावर प्लांट गोज डाउन द सिटी स्टॉप्स वर्किंग. सो सिमिलर है वैसी हाइपोक्सिया और इश्यूमिया भी. लेस O2 है अगर, तो लेस ATP प्रोडक्शन होगा. ओके, आता है केमिकल एजेंट्स, ड्रग्स, टॉक्सिन्स, हैवी मेटल्स एंड कार्बन टेट्राक्लोराइड. थर्ड नंबर आता है फिजिकल एजेंट्स, हीट, कोल्ड, रेडिएशन, मैकेनिकल ट्रॉमा. माइक्रोबियल में बैक्टीरिया, वायरस, फंजाई, पैरासाइट्स एंड इम्यूनोलॉजिक हाइपरसेंसिटिविटी रिएक्शंस एंड ऑटोइम्यूनिटी. न्यूट्रिशनल में प्रोटीन विटामिन डेफिशिएंसी और एक्सेस. सो रीड दैट, ये इतने पॉइंट इतने सारे, इट्स वेरी इम्पॉर्टेंट. ओके. Okay, now this reversible cell injury concept etiology is over. Then alright, let's move on to page 2, that is pathogenesis and mechanism. So pathogenesis में क्या है कि how does this damage actually happen under the hood? So there are 5 major steps you need to map out in your mind. That is ATP depletion, intracellular Na plus and Ca2 plus increase, anaerobic glycolysis and lactic acidosis, ribosome detachment from ER, and mitochondrial dysfunction. So first we are gonna read about the ATP depletion. So it always start here. Okay, pathogenesis is always start here from the ATP depletion, and hypoxia leads to decreased oxidative phosphorylation, which plummets your ATP levels. This, okay, and when the pump stops working, sodium, Na plus and water rush into the cell, which results to what? Cell swelling. So cell swelling, we can say that cell swelling is the earliest sign of cell injury. Okay, then comes intracellular Na plus and Ca2 plus increase. Because the pump failed, osmotic pressure pulls water inside. Crucially, calcium Ca2 plus rises in the cytoplasm. So think of calcium as a destructive light switch. Means it activates wild enzymes like phospholipase, proteases and endonucleases that start chewing up the cell component. Okay. Then third comes the anaerobic glycolysis and lactic acidosis. Okay. So the cell tries to survive without oxygen, so it switches to anaerobic pathways, and this accumulates what? This accumulates lactic acid, and which results into the less intracellular pH, and because of the less intracellular pH, it inactivates the crucial enzymes and causes chromatin clumping in the nucleus. Okay. Then comes the ribosome detachment from the ER. As the endoplasmic reticulum swells, the ribosomes literally drop off. So no ribosomes means reduced protein synthesis. You can you may know that ribosomes is responsible for the protein synthesis. So if no ribosome means reduced protein synthesis, and this causes lipids to get trapped and deposit inside the ER, which creating visible fatty changes, especially in major metabolic organs like the liver, heart and kidney. And then comes the last, mitochondrial dysfunction. So the mitochondria swell up and form a small amorphous densities. So this topic, pay close attention here for your exam, that this line, pay close attention, that small densities are reversible. It is very important for the NEET MDS exams. Okay, it is small amorphous density form means reversible stage. And if you see large, then calcium phosphate densities, the line has been crossed into the irreversible injury. Okay, if small density are still reversible, and calcium phosphate density that is large, it's from the irreversible injury. Now if we look at the cell under a microscope, or look at the organ as a whole, what do we actually see if we look at the organ as a whole and under the microscope? So let's check out the page number 3, that is morphology of reversible injury. Okay, so the gross appearance, that organ becomes pale, enlarged and heavy. To the naked eye, the organ become pale, enlarged and heavy, and if you cut into it, okay, if you cut this part, okay, the surface look moist and glistening. So look at the this drawing of the liver, okay, and this the classic exam buzzword here is a boiled meat appearance के जैसा ही आपको दिखेगा. Okay, then B, if we zoom in under a standard light microscope, we see a quadrant of classic features, that is 4 features, that is cellular swelling, hydropic change, fatty change and nuclear changes. Okay, in cellular swelling, the earliest change, cellular swelling is the earliest change of the cell injury, as we read above, and the cytoplasm look pale and granular. Hydropic change, also called vacuolar degeneration, you will see clear water-filled vacuoles inside the cytoplasm. Fatty change, clear lipid vacuoles clustering inside parenchymal cells like liver and heart. And nuclear changes last, chromatin clumps together, but crucially the nucleus remain intact, and if the nucleus break part or dissolve, it's necrosis, not reversible injury. If nucleus remains intact, it is reversible injury, but if nucleus breaks apart or dissolves, so it's necrosis, not the reversible injury. Mark that point, is very important. Okay, then we comes under the, if we go ultra high resolution with an electron microscopy, so look at the circular cell drawing on the bottom of the right, that this thing. Okay, we spot mitochondrial swelling with amorphous densities. Okay, and ER dilation with detached ribosomes, membrane blebbing, the cell membrane forms restored blotches, and myelin figures. We found myelin figures. So this 5 features what we see in electron microscopy of the cell, this will we see, and these are the most important part of the which comes in your exams. Then where comes in types of reversible cell injury. Okay, and we have cellular swelling, that is cloudy swelling, hydropic degeneration, a severe form where nucleus get pushed aside by water vacuole, fatty change, intracellular accumulation where abnormal substance like glycogen, protein or pigments accumulate like Russell bodies or lipofuscin. Intracellular accumulations and fatty change are the 2 most important high yield exam point which comes always in your exam, whether it is PDS second year exams or NEET MDS. Okay, so read it carefully, and I make a box for you students, high yield exam points. Okay, that most common cause hypoxia, ischemia, earlier chain cellular swelling, key organelle chain mitochondrial swelling, amorphous density. Key difference from irreversible membrane still intact, Ca2 plus fatty chain best seen in liver alcoholism, and heart is anemia. Okay, now deep dive into the cellular swelling. Let's do a quick deep dive into the most common manifestation cellular swelling on the page 5. Okay, as we established, it's an energy failure. Cellular swelling, it's an energy failure. So what is the core concepts? That the earliest and most common manifestation of almost all forms of cell injury results from failure of energy dependent ion pumps in the plasma membrane and leading to inability to maintain ionic and fluid homeostasis. So we look at the pathogenesis step by step. Okay, so injury leads to a drop in the ATP production, that is this Na plus and K plus pump fails. So Na plus accumulate inside, water follows the sodium gradient, and the cell ER and mitochondria swell up like a balloons and membrane blebs form. Look at the organs most commonly affected box on the right, that is this. Okay, so it's the liver, which is hepatocytes, kidney, tubular cells, myocardium, and the oral epithelium. So what is reversibility and the outcome, what we gain and this? So it is completely reversible if you get rid of the stressor because the membrane is still intact. But remember, if the injury continues unhindered, it crosses the point of no return into the necrosis, that is this. That fully reversible if stimulus is removed and membrane is still intact. If injury continues, then irreversible injury is formed, and then so necrosis is formed. Key hallmark, membrane integrity is maintained. Okay, then deep dive into the fatty change steatosis. The core concept is abnormal accumulation of triglyceride neutral fat within parenchymal cells, particularly in organ involved in fat metabolism. It occurs in organs that normally do not store fat. Okay, what causes this? Alcohol, the absolute classic. It disrupts fatty acid oxidation and VLDL synthesis. Okay, obesity, diabetes. In increases fatty acid delivery to the liver, causing a lipid overload. Okay, then protein malnutrition. If you lack protein, you can't make apoproteins, but without apoprotein, fat gets trapped inside hepatocytes, and because of this it has no vehicle to export it out. And then comes hepatotoxins and drugs. Okay, for your pathology lab exam, remember the special stains on the right, because routine HE staining dissolved fat, leaving empty with white spaces. We must use frozen sections with oil red O and Sudan IV on and fat disappear in routine HE paraffin sections. This is the lab exam part. Now if we go to the intracellular accumulation and oral pathology, so let's finally wrap up with page 7, intracellular accumulations, the last topic of this lecture. And this happens when a cell can't metabolize a substance fast enough. So we categorize them into 4 buckets, that is lipids, proteins, glycogen and pigments. So lipids, triglyceride, cholesterol like foam cells in atherosclerosis, or lipofuscin, the golden brown wear and tear pigment of the aging. Okay, so remember this 3 things, that is triglycerides part, the cholesterol ester part, the phospholipids part, which is example Niemann-Pick disease, and the lipofuscin part, that is wear and tear pigment, golden brown granules in liver, heart, neurons seen with aging. Now comes into the protein part. Protein part, what? Like Russell bodies in plasma cells, or Mallory-Denk bodies in alcoholic hepatitis. These 2 bodies are very important from exam perspective. Okay, the Mallory-Denk bodies and Russell bodies. Okay, and then third part comes like glycogen, seen in diabetes mellitus or glycogen storage disease like von Girk or POMP. We stain glycogen using PAS, okay, periodic acid skiff, which gives a magenta red color. And then comes from deep part pigments, exogenous like carbon tattoos, or endogenous like hemosiderin, melanin and bilirubin. Crucial section for dental and oral path student, that look at the right column, oral pathology relevance, that this is incredibly high yield for the dental exams, and it is very important for the NEET MDS perspective, that Russell bodies, you will see this plasma cell inclusions in chronic periodontal lesions, periapical granuloma and inflammatory gingival hypoplasia. Melanin accumulation, a normal physiological oral melanosis and amalgam tattoo or Edison disease, pigmentation on the oral mucosa. And the third part, hemosiderin, seen in a fibrous epulis or giant cell granuloma after a hemorrhage. And lipofuscin, aging changes inside the oral epithelium and salivary gland, SNR cells. So this will all complete the whole topic, reversible cell injury. That is a complete high yield mastery of reversible cell injury. And if you like the lecture, drop a comment below letting me know which part you like the most, and what I had to or upgrade my thing. So don't forget to hit that like button, subscribe to the channel for more simplified high-end notes, and you can message me on Insta. My Instagram handle is Dent Mindset, so you can message me for this notes, and I will see you all in the next lecture. Keep studying smart. Thank you.